Okay, so thank you, Angela, for the very kind greeting. So I will be talking to you about a little bit today about space, naturally. So being a neuroscientist, I've always been interested in the symmetry between space and the human brain. And in the same way that the universe is an orchestra of time and space, I've always seen the development of the human brain in the same way. So, for example, if you take a look up at the night sky, it's something I used to do when I was a kid. I'd go out and I'd stand on the front lawn and just be in absolute awe about the incredible beauty of our universe. And if you look at every single star, you can consider the fact that the photons are exciting your retina, have been traveling in some cases for thousands of years, and their final journey, or their journey ends in your eye. But the other thing that got me was just the staggering number of stars and the incredible size of our universe. And that's something which we struggle to comprehend. And so when you look at night skies that look like this, the number of stars is just astronomical. But when we consider what that represents in our galaxy, it only represents about that much, that tiny little red circle. That is everything we know. So that little red circle there gives rise to all of these stars. And so with that, you can see the sheer number of stars in the galaxy, which is just beyond our own comprehension. And in this case, it blew my mind when I studied neuroscience that the brain actually has more cells in it than there are stars in the galaxy. So as John said earlier, there's an approximately the same number of stars in the galaxy as neurons in the brain, but that's not considering all of the glial cells and the microglia and oligodendrocytes and other cells that allow our brain to work as one whole full unit. And in addition to this, it has over 1 trillion functional connections, which is just numbers which we can't comprehend. And the point of neurodevelopment is trying to unravel the mystery about how this incredible structure with all of its complexity arises from one single cell. So naturally, this single cell must have all of the information inside of it to generate not only the human brain, which is, as Brian said earlier, one of the most complicated things in the universe, but also to develop all of the different types of cells, all of the connectivity, to generate the morphological structures, for example, the cortex or the hippocampus or the midbrain. And it has to have all of the cells with the correct electrical activity. And so all of the information to generate this incredible structure is in this one single fertilized cell. And then this cell goes on a journey of time and space, where it experiences different developmental cues at different points. And that allows us, or allows the cell to generate into a full functioning human being. And to do this, it has to divide to form trillions of cells, approximately 30 trillion cells in the human body, not considering the cells that die and are replaced every single day. So what I want to do with you today is run through a few different stages of embryonic development. And hopefully you'll get a better understanding of how this single cell can be converted into a fully functioning human brain. So if we take a look at the timeline of neurodevelopment, there's a key few stages. So there's proliferation or neurogenesis. They're the same thing, and it's the mechanisms where cells divide to generate other cells which are fated to become neurons. There is differentiation, where these cells take on a very specific identity. For example, are they going to become an interneuron or a projection neuron? Will they become a retinal cell, or will they become a cell in the piriform cortex, which allows you to smell? These cells must also migrate. Cells primarily are born in proliferative regions of the embryonic brain, and they actually move through the brain to go to the place that they want to, or that they are fated to construct. There are periods of cell death. In embryonic stages, the cells are refined. These neural networks are refined by removing all of the cells which are not correctly integrated and leading them to die. And then lastly, there is synaptogenesis, which is the origination, or how all of these different neurons connect with each other to form massive incredible networks, which allows us to do things like smell and interpret and have consciousness and other things such as that. So the first thing we'll take a look at, and my personal favorite, which is proliferation or neurogenesis. How are these cells born in the brain? So I'm not going to talk in great detail about the very early stages of development, for example, gastrulation. And the reason is because it's not clearly linked to the brain at those early stages. We'll be talking about primarily the development from what's called the telencephalon, which is the anterior part of a ball of proliferative tubes, which ends up becoming the brain. So the first stage is proliferation. So if we take a cross-section of the brain, this is one hemisphere of the human brain, you will see a couple of distinct structures, but primarily this layer of darker brown is what's called the ventricular zone, and it contains progenitor cells, or cells which are actively dividing and generating other cells in the brain. And then these cells must migrate away from the ventricular zone and into other structures into the brain. So if we take a closer look at proliferation, and we follow this single progenitor, which is sitting right over here in the ventricular zone, this progenitor can divide through two different modes of division. So the first is asymmetric division, where it divides to form two distinct daughter cells. So the daughter cells are different from one another and also different to the original progenitor cell. And then these daughter cells can go on to either mature into a functional neuron, or it can become what's known as an intermediate progenitor, which undergoes symmetric division to generate two identical daughter cells. And the main difference, the main reason that these two modes exist that's currently known, is that asymmetric division allows early differentiation of cells. It allows early diversity to be generated, whereas the symmetric divisions are associated with amplifying the number of cells in a network. So if we take a look at what actually happens and what this looks like in vivo or in a animal or in the brain, this is a set of one developmental structures known as the MG, the structure that I study. And you can see, this beautiful, almost like a painting of neurons. And the cells represented in blue, these blue circle dots, are cells which are currently proliferating, and the green cells are cells which are moving and migrating away from the MG towards the striatum and towards the cortex to mature brain structures. And this is one of the most fascinating things that I've ever learned, but it's also an incredibly beautiful thing. And the beauty in looking at the brain, in my opinion, is equal to that of looking out into the incredible abyss that is space. So following proliferation, when we have these cells must take on their final identity. And differentiation is the process by which cells obtain their unique identity. And this identity is massively varied. So it can be associated with the connectivity. So you can have two cells with the same morphology and the same target cells, but have completely different afferent or presynaptic connections. They can have a different spatial location. They can end up innervating different structures. They could become a projection neuron or an interneuron or a glial cell. They can be born at different times. They can have different roles in the brain, for example. And differentiation, or the study of differentiation, is the role is to identify how these specific characteristics of cells are specified during development. And so we'll talk about three relatively canonical ideas of differentiation in the literature, and that is temporal specification, or time; spatial specification, which is effectively where in the brain the cells are; and then genetic switches, which effectively underlies the other two modes of differentiation. So first, temporal specification involves or is underlined by the fact that different cells are born at different times. And this is one clear example. During embryonic development, neurons are preferentially born earlier in embryonic development, whereas the glial cells, such as oligodendrocytes and microglia, are born later during embryonic development. And so clearly the time of birth predicates a cell to forming a specific connectivity pattern, or in this case, a specific identity. And not only that, there's also spatial specification. So I put this image up earlier, showing the MG and the LG. Now the MG and the LG are two embryonic structures, but they generate completely different cells. So for example, the LG primarily generates projection neurons, which are fated to innervate the striatum, or interneurons for the olfactory bulb, whereas the MG generates interneurons for the striatum and the cortex, but doesn't generate any projection neurons. And so you can see by having different things occurring at different times, in addition to at different places, you can start to build up this massive network of different unique cells. So the last, and probably one could argue the most important mode of differentiation, is genetic switches. So for example, a genetic switch is the simple either upregulation or downregulation of a gene at a developmental time point, which can completely alter its developmental trajectory. So for example, if we consider this progenitor cell, it may divide symmetrically to form two identical daughter cells. But these daughter cells, during their developmental trajectory, change and differ genetically. For example, the cell down the bottom may express or downregulate a gene which changes its identity, which may cause it to migrate to a different structure, or it could cause it to become an interneuron and not a projection neuron. So there's a massive amount of diversity here. And you can see how once you start taking spatial and temporal and genetics all together, you can start to build up the diversity of the brain. Now, third, these cells need to migrate towards their final structure. And migration is the physical movement of cells. This was something I was completely ignorant to until I studied it, which is that cells are born and move across the whole brain. And they do this following very specific mechanisms. So I will introduce this very briefly. And the mechanism is known as chemoattraction. And there's also chemorepulsive mechanisms in the brain as well. And so in this case, if we consider a premature or an early neuron, in its membrane, there will be membrane proteins. And these membrane proteins are ionically attracted to ligands, which are expressed in the structure which this cell is to migrate to. So for example, if we take a look on the left, you may have the neurons that are sitting in the MG express specific membrane proteins on their surface, and ligands that are present in the cortex in a gradient towards the MG. And so this cell, with its membrane proteins, is constantly attracted to the higher concentration of ligands and is dragged through the embryonic brain towards the cortex. And if you see this in practice, it's absolutely incredible watching these cells move towards their final location. One of the most interesting, and again, possibly the most counterintuitive period of neurogenesis or developmental neurodevelopment is the controlled cell death, or apoptosis. And it's known that in the brain, cells are massively overgenerated. And there's likely this is associated with redundancy. So for example, if you need to create 100 billion cells in a perfect order with a perfect orchestra of activity and connectivity, you don't want to simply just produce 100 billion cells. The easiest way to do it, generally speaking, would be to produce 200 billion and then get rid of all the ones which didn't make the cut. And then you're left with only those really strong, refined cells. And so this is exactly what happens in the brain. You have these stages of developmental apoptosis where the cells are refined, and that looks a little something like this. So if we consider a network of developing cells, a specific number of cells defined by their context will decide, because, for example, they're not integrated into the circuit enough, or they're not particularly useful, or they may have had genetic issues, enter a stage of apoptosis, and the cells and the network is refined. And so, as I alluded to earlier, this system is initiated by transcriptional switches. So the cells upregulate genes, which causes them to upregulate proteins, which cause the cell to kill itself or induce cell death. And the parameters that determine if this cell will undergo apoptosis includes things like how it's connected in the network, its membrane potential and its electrical activity, in addition to other factors such as its cell identity or its genetic program and things like that. Now, the last part of neurodevelopment is synaptogenesis. Now, this is by far probably the part of this which I'm the least well read on, but I have a decent understanding. We'll try and go through it a little bit. So when you have cells migrate into the final structure of the brain, they will produce what's called a neurite outgrowth, which is this little projection here. And this continues to grow and it generates what's called an axonal growth cone, or in this case, an axon, which was talked about earlier, the thing which sends the signal to the other cells in the brain. This growth cone is attracted to specific postsynaptic boutons or specific cells and the dendrites on those specific cells. And so once this occurs, you end up with this generation of a fully functional connection. And if we take a closer look at this functional connection, you can see that this axonal growth cone converges on the postsynaptic dendrite. You assemble a synapse and integral membrane proteins adhere the postsynaptic cell to the presynaptic cell. And you lead to the generation of a mature synapse, which allows the electrical transmission of signals throughout the brain. And so from these 5 very, very complicated, but also incredibly fascinating steps, you can start to build up the incredible diversity of cells in the brain, and you can start to piece together how networks are formed and how mature structures are specified over development. And not only that, but also understand why at specific developmental stages, you don't have the brain resources that you do to do specific tasks. For example, you wouldn't throw a cricket ball at a 6-month-old baby. They're probably not going to be able to catch it. And by studying development, you can actually understand the brain state at these different developmental points and understand how more complicated behaviors are developed over time. And so with that, I'd love to take any questions if you have any. And I really hope you enjoyed the talk, and I hope you learned something.
Thank you so much, Rhys. That was very interesting. And I'm sure that you had a few very interesting questions out of that. Please do ask, feel free to raise your hand if you have a question for Rhys, or you can type your question into the chat. Just to start things off, I can't see any questions there just yet. I'm just wondering, Rhys, how are these processes so precisely timed within the brain, and how does the brain sort of send signals to allow these things to occur at such precise developmental time points? And sort of in extension to that, are there instances in which this process is maybe delayed or happens at the wrong time point, and what might be the outcome in that case?
Yeah, so that's a great question. So in the case of how the timing is specified, this is currently somewhat debated, because coming back to that first fertilized cell, that cell must have all of the genetic information and all of the information required to elicit those developmental changes 6 months later, or a year later, or 2 years later. And so primarily, I think the current understanding, which is likely to change at some point in the future, is that it's underlied in almost entirely by genetics, and that the cell is predetermined to undergo a specific developmental switch at a specific time based on its genetic profile in the fertilized cell, and then also at different developmental time points. And examples of when development is delayed, absolutely, there's a number of different conditions that arise when development is delayed. Or in this case, even a developmental switch is premature. So a good example of this is severe autism spectrum disorders have been associated with early maturation of cells and overproliferation of cells. And so that's an example of where you have a change in the embryo, a different developmental switch being elicited, leading to a completely different change or a completely different brain structure later down the track. I hope that answers your question.
Yeah, it does. That was very great. Thank you very much. Very interesting stuff. So now we do have a couple of questions that have come in from the audience. So Sarah is wondering what happens to old or rejected cell material in the process of refinement.
That is a great question. And my understanding of this is that these cells are broken down. So for example, talking about the glial cells in the brain, you have things like your microglia and your oligodendrocytes, which facilitate in effectively taking the genetic and the protein materials and that from the cells which are dying, and it is reconstituted into useful things that the brain can then use. It's not like it's just gotten rid of as waste. Our brain has the ability to reuse things that it breaks down. Hope that answers that. Great. Is the brain connected with this space through any kind of energy or something? I wish I could answer that question. Like, I wish I could answer that question, but I have no idea. Look, all I know is that pretty much everyone at some point in their life looks up to space and is just absolutely gobsmacked with it. And maybe it is linked, but you know, I don't know. I wish I did.
It's a question for another day, perhaps, Rhys.
Exactly. It might be a question for our ancestors, not our ancestors, our great grandkids. They might somehow figure out a way to study this.
That's
but currently we don't. No, it's not discovered yet. But it's a great question. It's a great question. I think the problem with something like that is it's not so much the question itself, it's that there's no great way of actually investigating it. So if you could come up with a way of studying that and you could prove that there is a connection between the brain and space, guaranteed you will win a Nobel Prize. Like 100%. So if you want to give it a go, absolutely go ahead.
Watch this space.
Exactly. Exactly.
So we have another question, Nick. Are these the mechanisms that also underlie neurodegenerative disorders?
Absolutely, Nick. That's exactly right. So when these neurodevelopmental stages are perturbed, you can induce disorders. So for example, I mentioned earlier that autism spectrum disorders, the severe cases, are associated with change proliferation, but it's not just proliferation. When you change one stage, it leads to a follow-on effect. So for example, if you generate twice as many cells in a structure than you're supposed to, you then suddenly have twice as many cells migrating, and you have less resources for each of those cells, and you have the integration into the circuitry will be different. And so the specific stages that we talked about in the talk, each of them is associated with distinct disorders. So for example, proliferation is associated with autism spectrum disorders, whereas things like Parkinson's disease and Huntington's disease are associated with other neurodevelopmental conditions. And things with Parkinson's and Huntington's, interestingly enough, there is some evidence which is right on the forefront of our knowledge at the moment that it's actually induced in the embryos. So you're predicated to these conditions, but that's something which still is it's in its infancy and has decades of research which need to be done to be able to see the exact mechanisms which underlie it. But yes, you're exactly right.
Right, okay. Well, it seems as that's all the questions for the time being. Thank you very much for answering those, Rhys, and for
No
your
worries.
presentation. We much appreciate.